out here at the conference a couple of hours ago. I'm going to talk about this really challenging issue about what is a healthy quarter. And this will drive discussions around the world about where we really should be setting somatic cell count levels for healthy milk. And in the United States has undergone quite a bit of criticism because its somatic cell counts that are allowable are much higher than is achievable in many other countries. So if you look at the history of the literature going back to the 1930s, people sort of have looked at it and said, well, it looks like a somatic cell count about 100,000 and less indicates healthy secretion, and above that is an abnormal secretion. What is interesting is this has been done purely by an eyeball, and yeah, that looks about right, and yeah, I sort of agree with that, rather than using quantitative statistical techniques. So our objective was to apply segmented regression to objectively determine the somatic cell count cut point to differentiate healthy milk from abnormal milk. And this is about one of the first applications of this technique in clinical veterinary medicine. It has been used somewhat in nutrition studies in animal science. And the first publication of its use, I believe, in veterinary medicine, clinical veterinary medicine, has just come out this month by Dr. Treffs. I'm a co-author on that paper, and he'll be presenting an abstract on the application of that for hyperkalemia tomorrow morning. So we'll go back to probably one of the seminal studies on this, which came out of Germany by Toll, originally in abstract form in 1971. But here's a schematic of it in 1982. And what this is supposedly showing, sorry if I can get this to work, is that at a certain somatic cell point that various constituents change. So if you can pick this one up here, which is potassium, you'll see that it starts to decline after a certain cell point. Likewise, if you look at chloride, it appears to go up, and sodium appears to go up. And again, this general consensus, if we asked you in this room, you'd go, yeah, we probably think it's somewhere around 100,000 is where these constituents of healthy milk seem to change. And why are they changing? Because there's either increased permeability or interference with actually the production of milk. Okay, this is schematically presented in a different format in Veterinary Medicine 11, the textbook, which is really a redrawing of a presentation by Haman, who's done a lot of work on this and has been very passionate about this. This was from the World Buiatrics Congress in 2002. He went ahead and took the mean value up to 100,000 and then log transformed it. So if it stayed at 100%, it pretty much stayed at zero. And again, his argument was that at about 100,000, things started to change and go up and down in different directions. I think both of these graphs would sort of suggest that it's a little bit after 100,000. And then if you look at this from a normal distribution, he hypothesized that a healthy quarter probably has a mean count somewhere around 60,000. And then if that was not inflamed, most likely due to some type of infection at some time, but remember somatic cell count's an indication of inflammation, that this would drop down to about 100,000. So there's a lot of lines of evidence to say that's a pretty good number. So what did we do in our study? We had a unique data set from one herd where we applied a lot of potential tests for subclinical mastitis. So this analysis right now is based on 438 quarters from 112 dairy cows from one farm at the University of Illinois. And we measured somatic cell count here using the D. Laval cell counter, which has been extensively validated as being accurate up to about 300 cells per mil, but probably has a little bit higher coefficient of variability than some of the other methods for looking at somatic cell count. And we looked at 8 tests that potentially have been associated with inflammation, and you can see them listed there, and we're going to talk more about them here in a minute. So the statistical analysis that I believe is novel is called segmented regression. Other terms for it are broken line or piecewise regression. And then this is not quantile regression or cubic spines. The focus of this analytical technique is we want to identify where one particular modeling relationship changes to another particular modeling relationship, and we're really interested at that breakpoint and identifying a 95% confidence interval for that estimate. If you do quantile regression or cubic splines, what you do there is say, we're going to do 100,000 as the breakpoint and then model before and after that. That's not the question we're interested in here. We want to know is when does a secretion from a milk, from a quarter look like it's abnormal? Okay. So we're doing a complicated statistical approach. I've worked with nonlinear regression for about 20 years. It's one of the hardest statistical methods to get used to, and particularly when you do it with a nonlinear mixed approach, because now we have 4 quarters within a cow, so that nesting has to be taken into relationship. Certainly it's easier if you just do it as nonlinear regression. Both of these are in SAS 9.4. I'm not going to put the code up there because it's quite intimidating, but I'm happy to share that with any of you that are passionate about nonlinear regression. Anyway, the secret to this is that you continue to move this breakpoint and you try and minimize the sum of squares for error over the entire fitted equation. And what you hope to do is when you're on this side, it starts to go down, then we call it hitting the bottom of the bowl, and then it starts to go back up again. And we want to get this minimum point that optimizes the distribution and is therefore the breakpoint that we're trying to identify. So let's look at those 8 potential analytes and see what numbers they come up with and how close we are to our 100,000. So this is in order. So this is the lowest one detected. We're fitting a straight line with no slope on it up until this breakpoint, and then we're fitting another linear line, and somatic cell count is expressed as log to the base 10 and expressed logarithmically like so. And you can see here is the breakpoint at 98,000 cells per mil, and here is the 95% confidence interval, which spans quite a bit. And this was measured by the Hariba potassium meter, and we've been working with the Hariba meters for the last 5 years. The second most sensitive one was lactose. The breakpoint here was 112,000. The confidence interval was a little bit narrower. And we used a modification of a technique described in Journal of Dairy Science, where we provide lactase solution to milk and then use a glucose meter to measure the lactose percentage. This worked very, very well for us, and it's a low-cost method of getting lactose percentage. The third one was sodium, with a breakpoint of 114,000. As the milk blood permeability increases, sodium moves from plasma into the mammary gland, so we get an increase in the sodium concentration. And we measured that one using the Hariba sodium meter, and we've validated that for measuring sodium in milk. The next one was milk esterase test using what's called a porter check unit with a reader, where you actually look at the color that is consistent and linearly associated with the esterase activity, and then this gives you a more objective measure than just 6 categories right here. You'll see its breakpoint was 148,000, and again was constant and then went up in a linear manner. Milk calcium, ionized calcium. This is very problematic because it's dependent on milk pH, and as you'll see in a minute, milk pH goes up with increased in somatic cell count. That's been known for quite a while, but we use the Hariba calcium meter for this. and we were able to detect a breakpoint, but you can see the confidence interval is very, very wide just because of the large amount of scatter in our samples. And that's in everyone's data who's worked with ionized calcium. It's to do a lot with the amount of casein that is in the secretion. Milk pH, we've got a paper in press in Journal of Dairy Science on this, but you'll see here again that milk pH has a lot of scatter but is relatively constant up until a break point of 177,000 and then goes ahead and increases. And then a technique that has been around since the 1970s. The overall conclusion of this technique is it's not sufficiently accurate to detect subclinical mastitis, even though it has some predictive ability. Its breakpoint was 189,000 for us on our analysis, and we measured that using a Hariba conductivity meter right there. And the final one was a SomaticelTM modified Wisconsin test. The SomaticelTM really goes back to tests that cause increased viscosity, such as the California mastitis test. And its advantage is that it tries to do that in a more quantitative manner. You can see with our data set that the actual confidence interval is incredibly narrow for the breakpoint, but the breakpoint was the highest on this, and we published on this recently in Journal of Dairy Science, in that the SomaticelTM test as we used it, which was not consistent with the manufacturer's recommendation, did cause some challenges with the interpretation under about 800,000 cells per minute. So in conclusion, let's look at this as a summary. Here is the breakpoint for each one of these changes. Here is the 95% confidence interval. So our data for the first time using objective quantitative analysis would say that 100,000 looks like a pretty good data point for a breakpoint for a healthy quarter. Now, what I would really like to see is those of you in the audience that have these huge data sets with 10,000 or 50,000 cows or quarter samples, it would be interesting to apply the same analysis to this particular thing to see how robust we can make this 100,000 estimate. Because if we can make this robust, I think that gives us all a lot of energy in having this as our next big guide, in that we really should see quarter somatic cell counts at 100,000. Thank you for your attention.